here. So welcome everyone to our College of Marin Community Hour. I'm Paul Daubenmeier. I'm the STEM Learning Community Coordinator here at COM, also a chemistry faculty, and we're so glad you're with us today for our talk today. We're hosting Dr. Emily Fox from our biology department, and she'll have a wonderful discussion for us today. But before that, I just wanted to say for a couple minutes something about our STEM learning community. We have a Canvas page, and for any students who'd like to join or dig around a little, we have different opportunities that we've presented. I'll drop the link in here. It's right there in the chat. And I'll share our website as well, just to introduce you a little bit to our virtual presence. Our physical presence is downstairs on the first floor of the SMN building next to the math lab. If you've ever been down there, there's a space there for studying, working, meeting with faculty, as well as social events. And this is our digital page right now. So you can see we've been hosting a speaker series all semester. And if you want to go back and catch any of the speakers, if you've missed them, we start off with Dr. Farisa Morales. She's presenting her journey to go from community college to the Jet Propulsion Lab at NASA. You can click up here and get the recording for that. So this talk will also be recorded. So we're going to encourage folks to reference that and share it to help build the community. We also have scholarship opportunities, summer internships that are a range of disciplines, as well as links to on-campus or virtual tutoring through the Math Lab, through the Tutoring Center, and then a way to connect students together through digital whiteboards. So we encourage you to click in, join the community, and click around. And let me know if there's any questions. My email address down here, I'll drop it into the chat as well. I know some of you in this meeting, but some of you we don't know. And then also want to just share with you a little testimonial from one of our students who graduated last spring, but she made a little video for us that I think is pretty representative of the experience that students have with the learning community at COM. So give me 1 second here and I'll share this video with you all. So let's start this up. And some of you might recognize Oda. Here you go.
Hi, my name is Olaiklet Piccinini, and I was a College of Marin student for the summer of 2016 until the spring of 2020. And I am majoring in cognitive science and minoring in data science. I'm here today to share my experiences as part of the STEM learning community of College of Marin and to tell you what you should expect after joining. As part of the community, I joined study groups and connected with students. I attended to scholarship presentations that taught me how to apply to these scholarships and helped me land a scholarship of $30,000 to cover my academic expenses. I was mentored by faculty members that helped me take actionable steps towards my end goal of becoming a data scientist, and they gave me career guidance and were interested in my goal. So I was provided with a list of summer internships to which I applied to after my mentor helped me review my application, and I was able to get an internship with the Northern Arizona University, a paid internship. I was able to learn how to code in Python. I was also able to use data query and discovered the provisional shape of an asteroid that goes around the sun every 319 years. Being in this community allowed me to hear stories from women in science through the STEM speaker series and also through the collaboration of the Women in Tech. This community is an absolutely incredible resource for you and your STEM career. And if you want to learn more or are curious about it, please do not hesitate to reach out to Dr. Daubenmeier and Dr. Kukiata and take an opportunity to be part of it.
I'm very happy to share that testimonial from, again, one of our past STEM Learning Community students, and looking for opportunities to always build a digital community, as well as looking forward to the fall when we even will have some opportunities to see one another face to face. So just putting a plug in for the STEM Learning Community. I'll put the link in the chat one more time. If you're interested in applying, fill out your information. It's a quick little link, and then from there we'll be able to follow up with folks and try to match you with mentors, or also with students who are the same career trajectory as you. So I'll go ahead and pause right here and transition to Dr. Emily Fox. She's actually a new hire to the College of Marin. She's in our biology department, and she graciously offered to talk a little bit about a topic that's really obviously been in the news and controlled our lives for the last year. What's new with COVID-19 is the title of her talk. So she's going to share a little bit about her journey, and along the way, we hope we can use the chat function to open up some questions. So as she's speaking and giving a little intro presentation, obviously you can't raise our hands the way we normally would in person, but use your chat box. You can find it in the bottom of your iPhone. Sometimes clicking the more button opens that up. And feel free to ask a question. And after her talk, she'll be happy to lead us into her experience as to how she got here today, as well as any of the questions that might come up as she's presenting. So without going longer, I'm going to go ahead and hand it over to Emily Fox and let her introduce herself. Thank you for joining us today.
All right. Hello, everyone. Thank you so much for coming today. It's exciting to see so many student and faculty faces, and I'm excited to talk about this very timely topic. I have a background in microbiology, and I'm teaching microbiology here at College of Marin. So this is kind of an exciting time to be teaching microbiology when all of a sudden everybody's really interested in microbiology. So that is a great place for me to be. Today's talk is called Going Viral, What's New with COVID-19. And I am going to talk a lot about COVID-19 soon, but part of this speaker series is also about just learning the trajectory of how people ended up as scientists. So I'm going to talk about my path in how I became a scientist first, and then we'll come back around to COVID-19. So how did I get here? Well, I did not start out as a scientist. I started out as an artist. When I was in high school, I went to San Francisco School of the Arts. The picture of me looking rather dramatic. I was in the theater arts program. Didn't really know that I was interested in science, and the science classes at the art school were not really the focus. So it wasn't until I started attending community college that I actually discovered that I had an interest in science at all. And I went to City College of San Francisco, where I found an incredible mentor in a biology teacher by the name of Krima Pogue, who really got me excited about all the tiny things that are going on inside of cells and in the microbial world. I initially thought I had an interest in nutrition, and so I made a little bit of a mistake in my educational path where I transferred to San Francisco State University to study nutrition. But I got there and I spent one semester there taking nutrition classes and realized this is all wrong for me. What was I thinking? And so I went back to City College and said, okay, I have to reevaluate. I really liked this kind of molecular microbial world that I've been learning about in these science classes. Let me try that instead. And so then I took more science classes to prepare for a transfer to UC Berkeley. And I studied molecular environmental biology while I was there and started doing a lot of research at the laboratory bench in a plant genetics lab, where I found another fantastic mentor, Yen Zhang, who really taught me how to actually do science with my hands in a laboratory. And that inspired me to go ahead and get a PhD in molecular biology. And so I did that at UCSF, where, common theme, I found an amazing mentor, Clarissa Nobili, who was not the head of my lab. She was in the same lab as me, a little bit more advanced, but she was a superstar and really pushed me to become a stronger scientist, was always giving me more things to do, more papers to read or review, or telling me that I should try this, or setting up collaborations, really helped guide my journey as a scientist. And she now has her own laboratory at UC Merced. And so this was my path. And on the surface, except for San Francisco State, it might look like a pretty straightforward path. I went to community college, I transferred to Berkeley, I got a PhD, bam, I'm a scientist. But probably like many of you, I did face a number of challenges along the way. And the first of those challenges was money. I grew up with a single parent. We were very low income. There was no money for college. And I worked the whole time I was going to school. I worked at Trader Joe's for 8 years, paying my bills in the Bay Area, which isn't cheap, right? Went to school part-time for a lot of my education so that I could make it through. And then the next thing that happened was in my early 20s, my mother was diagnosed with cancer. And so that was a really emotional time of figuring out how to support her at the same time as going to school. Very fortunately, she survived cancer and is doing very well today. And then the third thing that happened was one of the major challenges happened in graduate school, which is where my partner and I decided that we wanted to have a baby. I was an older student and I was starting to get a little worried about that biological clock running out. I had fantastic health insurance as a graduate student. And so we decided to go ahead and try to have a baby while I was still earning my PhD. And so that happened. And there were some challenges that came up for me during that around support from my advisor. You know, when you're in a PhD lab, your primary focus is supposed to be on your studies and your research. And so I had to deal with some comments and things around the pregnancy and birth of my child. But despite those challenges, I did finish my PhD when my first child was 8 months old and made it through that. So those are just some of the things that I overcame during my education. I'm sure a lot of you have really similar things going on in your lives now or will in the future. And so I'm going to pivot from here, from my story of how I became a scientist, into the COVID-19 part of the talk, unless anyone has any burning questions. Let me see, where's the chat? Okay, no burning questions yet, but if anyone thinks of anything, go ahead and put that in the chat and I'm happy to answer as we kind of go along. Here we are, on to the virus. So I just wanted to define some terms that I'm going to be using about this virus before we get going. You might have heard the term COVID-19. That refers to the actual disease, right? What you experience when you're sick. And the virus itself is called SARS-CoV-2. And so I'm going to be using that term a lot, SARS-CoV-2, as we go along to refer to the virus. So let's take a look at this virus. On the left here, we have an image taken under the microscope. This is an electron microscope showing 3 coronaviruses. And you can see they have this kind of round shape with these little sort of spikes coming off of them. And scientists who first saw these under the microscope said, oh, that looks sort of like a crown. And so that's where the term corona came from. Corona means crown, and so it's the crown virus. And if we look on the right-hand side here, you can see this cartoon of the structure of SARS-CoV-2. We'll start on the inside here. This squiggle is RNA, which is the molecule that contains genes that tell the cells that are being infected how to make more virus. And the little blue circles on the RNA are proteins that are just helping stabilize that RNA and package it together. The purple circle around that is a piece of membrane that the virus steals from the cell that it's infecting as it's leaving, and it makes this little protective envelope around the virus. And then around that, these kind of turquoise blue spikes are proteins that are really important for how the virus is able to get inside of our cells. And we can zoom in on one of those spikes. Looks like this. This is a sort of a chemistry model showing the shape, the actual shape of the proteins. And this little portion here is the part that is key to getting inside of our cells. The spike proteins bind to a receptor found on the surface of our cells called ACE2. It stands for angiotensin converting enzyme 2 receptor. And you can see a little chemistry model of that shown in brown here, and sort of how they fit together and interact chemically, allowing the cell to let the virus in. So let's look and see, how does that happen? How does the virus actually get inside of a cell, and what happens once it's in there? In this little cartoon, this big green box taking up most of the image is representing a human cell. This could be, for example, maybe a respiratory cell inside of the lung tissue. And this yellow circle is the nucleus, where the DNA of the cell is kept. Here's the endoplasmic reticulum, which is this membrane structure that has these purple circles on it called ribosomes that make proteins. And those will become important in a moment. On the outside of the cell is this ACE2 receptor that I mentioned before. Okay, so here is my little cartoon of the SARS-CoV-2, our little virus, with green spike proteins sticking out from it. And the first step is going to be binding to the ACE2 receptor. So it will come along, and one of those spike proteins will actually fit into that ACE2 receptor, sort of like a key sliding into a lock that's going to open the door to allowing the virus to enter the cell. So the second step here is entry. And you can see it comes in inside this little bubble, and then the genome, the RNA, is released from the virus into the cell. That genome gets uncoded, all the proteins get stripped off of it. So now you have this long strand of RNA that is ready to tell the cell how to make more viruses. It's going to turn the cell into a virus factory. There are two things that need to happen. The first is the RNA tells the cell how to make the virus proteins. You have translation of the viral proteins, so it goes to one of these ribosomes, which starts making the virus proteins. You can see these little spike proteins being made up here. The second part is making copies of the actual RNA so that those can be packaged into the new viruses that are being put together. Okay, and then the viruses are going to be assembled and packaged inside this bubble of membrane, and then finally released through the cell through a process called exocytosis, where that bubble of membrane fuses with the edge of the cell, releasing the virus outside of the cell, where it can go on to infect new cells. And it'll do this over and over again, producing many, many viruses and releasing them. Okay, so that's a little bit about just the biology of SARS-CoV-2 and how it works. Let's move on to prevention. And I wanted to hear from you, because I think we're all kind of experts on this now. What are some ways to prevent COVID-19? So you can type your answer in the chat box, or you can just unmute and share with us verbally. Either way is fine. Yep, I see a lot of people saying mask, 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 wear a mask, wash your hands, social distancing, wear 2 masks. Okay, yes, it sounds like we have all been told what to do in terms of prevention. Yeah, keeping your immunity strong, excellent. All right, so let's start with looking at masks. Do masks actually work? Why are all these scientists telling us to wear masks? So there were some scientists that did a really cool experiment that just made so much sense to me, where they took 2 mannequins of people and stuck them inside this box. And then they made one mannequin the spreader, that they were going to feed virus into the spreader and kind of spew it out into the air. And then they made the other mannequin the receiver, and they were just breathing kind of in and out through this artificial ventilator. And then any virus that gets breathed in gets collected in this little filter inside the receiver. And they can see, by putting different masks on the spreader and the receiver, do the masks work? And so here's a slide that, I apologize, I know it's a really busy slide with lots of data on it. I'm not going to go through all of the data on here, but I'll walk you through a little bit what this data shows. So let's look at just panel A here. You can see at the little cartoon on the top, here's the spreader on the left and the receiver on the right. And in this case, there aren't any masks yet. They're just looking at distance. If you move the spreader and the receiver farther apart, does the receiver get less virus? And the way they are measuring virus is shown in these blue and brown lines. The blue lines are measuring active virus that can actually infect cells, and the brown lines are measuring all virus, whether it's active or not. So it's just two different ways of measuring the virus that makes it into the receiver. So this is at 25 centimeters, 50 centimeters, and 100 centimeters apart. And this little drop looks kind of small, but this is a log scale. So that's actually a pretty significantly large change to move just from there down to there. So you can see as the mannequins get further apart, there's less virus making it from the spreader to the receiver. Panel B, now they put a mask just on the receiver, and they looked at a number of different types of masks: cotton masks, surgical masks, N95 masks, and fitted N95 masks. And what you can see is that the fitted N95 masks were the best at protecting the receiver. Panel C, now just the spreader is wearing a mask. And you can see when the spreader is wearing that fitted N95 mask, that blue line just completely drops out. That's the active virus. So no active virus made it from the spreader to the receiver when the spreader was wearing an N95 mask. And actually you can see the difference between the spreader wearing a mask versus the receiver wearing a mask is pretty significant too. That's why the news and the scientists have been telling us that it's really important for us to wear masks to prevent spreading it to other people. And then D and E is looking at some different combinations of a spreader wearing a cotton mask, and then this receiver wearing various types of masks, or panel E shows us the spreader wearing a surgical mask and the receiver wearing various types of masks. And so by looking at that data, you can just get a sense for which masks are the most protective. So I just thought that this is a really cool paper for seeing, oh yeah, masks actually work and you can actually measure that they work. So if you have know anyone who's been resistant about wearing a mask, maybe this is something you can show them. All right, so the other way that we have been learning about preventing is vaccination. Yeah, and I think a few of you mentioned vaccination as a way of preventing COVID-19. And there are 3 vaccines that have been approved in the United States: Pfizer, Moderna, and Johnson & Johnson. And despite the differences between them, they all actually work in a really similar way, which is through introducing the SARS-CoV-2 spike protein into the body. So there's just a slightly different cartoon of SARS-CoV-2 with the spikes shown in red. I'm using this one for a little bit. All right, so how does this vaccine work? In your body, you have immune cells that are just waiting to see if anything foreign comes into the body, like bacteria or viruses, for example. And they have special receptors that can detect those foreign things if they come into your body. And so when the vaccine comes into your body, that makes the spike protein in your body. And that spike protein will bind to one of these receptors and activate your immune system. The immune system is going to have 2 basic ways that it's going to become activated. The first is making antibodies. Antibodies are Y-shaped proteins that are shown in green here, and they can stick now to the spike proteins on the outside of the virus if the virus ever shows up for real. And the antibodies are going to do 2 basic things. The first is they surround that virus and kind of block all those spike proteins, so it's harder for the virus now to bind to a receptor on a cell and actually get inside of the cell. The second thing is that the antibodies target that virus for other immune cells to come along and eat them. It's kind of like playing that old game Pac-Man, where the immune cell comes in and it sees the antibody, and the antibody says, this is a bad thing, you should eat it and destroy it. And the immune cell says, okay, eats it up and destroys it. The second way that your immune system gets activated is by activating these special types of immune cells called T cells. When a T cell becomes activated, then it is going to patrol your body and look for any cells that have been infected with the virus. So here's your infected body cell over here. Oh no, it has SARS-CoV-2 inside of it. And one of the really cool things about all of our body cells is that they're constantly just taking samples of the molecules that are inside of the cell and displaying them on the surface so that the immune cells can check and see, is this just a normal good thing, or is this a bad thing that the immune system needs to deal with? And so when your body cell gets infected, it will take pieces of the virus and display them on the surface. So you can see that spike protein being displayed on the surface. And then the activated T cell will come along with its receptor. It'll detect that that spike protein is on the surface of the cell. And when that happens, the activated T cell will kill your infected body cell. And that's good because this has become a little virus factory, and we want to make sure that that body cell gets killed so it doesn't make more virus, and that will help halt the spread of the infection. All right, so the vaccine was just the spike protein setting off these two pathways, making antibodies and activating T cells. And then if the actual virus shows up, then the antibodies and the T cells can deal with the virus. And what the immune system does after the vaccine has come along is it will form what are called memory cells. And these memory cells are the ones that will recognize SARS-CoV-2. And they're just kind of saved in your body in case the actual virus ever shows up. If the virus actually shows up and enters your body, now those memory immune cells will really quickly make antibodies and T cells to fight off the virus. So here comes the virus, and the memory cells will just chase that out of there before you even knew that it was in your body, typically having no symptoms at all. Right, so that was sort of a general way that all three vaccines are working. Now let's look in a little bit at the specifics of Johnson & Johnson versus the Pfizer and Moderna ones, which are made out of mRNA. We'll start with Johnson & Johnson. So the way the Johnson & Johnson vaccine works is it provides DNA that encodes spike protein. So if you look at the virus, this is the SARS-CoV-2 virus. Inside that virus is the RNA, remember? And this little red part is showing the gene that encodes the spike proteins. And so what the Johnson & Johnson vaccine does is it cuts out just that gene and sticks it into another virus called adenovirus. Adenovirus, there are many different types of adenoviruses. Some of them cause the common cold, but this one in the vaccine will not give you the cold. It's been modified, so it has lost most of its properties except for the ability to deliver DNA into the cell. So the adenovirus will bind to the human body cell and come inside, and then it'll deliver that DNA to the nucleus of the human body cell. And that DNA now, because the scientists put the spike gene DNA in there, will deliver that spike gene DNA. Once it's in there, the human body cell will take that spike gene DNA and transcribe it or convert it into what's called messenger RNA. And then that messenger RNA will leave the nucleus. And here it is, the spike gene messenger RNA is here now, and it's interacting with a ribosome and getting turned into the actual spike protein. So what the vaccine is doing is it's providing the instructions to our body cells on how to make the spike proteins. And the cells themselves start actually producing these spike proteins. The spike proteins get displayed on the surface of the cell, and now our immune cells can come along and detect that those spike proteins are there and set off that immune response that we were talking about earlier, making antibodies, making activated T cells, making memory cells to save for later in case the actual virus shows up. Okay, so the Pfizer and the Moderna vaccines provide messenger RNA that encodes the spike protein. So it's really almost exactly the same thing. Let me go back here for a moment. What the Johnson & Johnson gave us was this spike gene DNA. What Pfizer and Moderna are giving us is just the spike gene messenger RNA. So it's just one step further along the pathway to making the spike protein. So in this cartoon, the RNA looks a little bit different. It's this purple cartoon here. And the vaccine is made by packaging that RNA into a little bubble of lipid. It gets injected and then it ends up in your cells. And once it's in there, it doesn't even need to enter the nucleus. It just gets it gets read, translated into protein. So the proteins are made here, and that then sets off the immune response, just as we saw previously. Okay, the last thing I'll talk about, and then I'll start addressing questions, is this idea of variants. So the variants are something you might have heard about in the news. They're kind of different versions of the virus. These are SARS-CoV-2 viruses that have mutations in the spike proteins. So the spike proteins have gotten just a little bit different in their chemistry, which makes them interact with the cells a little bit differently. And so far, the main effect of some of these variants that are of concern is that it's causing the virus to be able to spread more quickly. And the good news is that so far, the studies have shown that vaccination is still mostly effective against the variants. It definitely provides some protection. For some of the variants, some of the vaccines might work a little bit better than others, and they might be slightly decreased in efficacy, but so far, definitely still worthwhile to get the vaccine. You're getting at least partial protection against all of the variants that exist now, and it's really important for all of us to get vaccinated as quickly as possible before more dangerous variants can arise. Okay. So I am ready for questions. I saw a few pop up here, so let me go back into my chat first. I see.
Yeah, the chat's been exploding, Emily. This was an excellent presentation with visuals and detail that no one's ever heard before. So,
okay, great.
Yeah.
And if any of that biology lingo lost anybody, feel free to ask really basic questions about like, what did you say? What does that term mean? Because I know we're a mixed audience. Okay, so cotton masks are more comfortable. Is it better than surgical? I noticed surgical makes me have more mucus or sweat. So both definitely work. We could go back to that data slide for a moment. And oops, both definitely work. It looks like surgical masks are maybe a little bit more protective than cotton masks, but they're not that different from each other. And again, this is data that was collected in a laboratory environment with this very controlled setup with the two mannequins, which gives us really nice controlled data, but it's not going to exactly replicate what happens in the real world where we have, different kinds of wind and different distances between us. And so if you are more likely to wear a mask, if it's a cotton mask, then go for it. Do the cotton mask is better, definitely better than nothing. And the surgical mask, this data makes it seem like it's maybe slightly more effective, but it's not a huge difference. So I think that it's okay. It's okay to choose what's most comfortable for you. I see, should we always have been wearing 2 masks, or are we supposed to wear 2 masks now because of the new variants of COVID-19? Yeah, so it's tricky, and we're always still trying to figure that out. Remember, initially they told us not to wear masks at all because they were worried about the supply for healthcare workers. And then the recommendation went to 1 mask, and then it went to 2 masks. And the idea behind 2 masks is just that you have that much more layers of material between you and the air. But again, there's always going to be a trade-off between what are you actually going to wear. If people won't wear something that's uncomfortable, then it's best for them to wear something that is more comfortable so that they'll wear it continuously. It's the most important thing is that you wear it all the time. And so that put the priority there. Okay, it says, do you know if people that received the vaccine and got COVID-19, can they still spread the virus? I'm just gonna go out of here for a second. Yeah, so this is definitely a question on scientists' minds. There are some new studies in healthcare workers that have been trying to get at this question. The original studies, they were only really measuring symptomatic infection. So they didn't know the answer to that very well, but there's some new studies indicating that even asymptomatic infection is drastically reduced by the vaccine, down to, I think the percentage, if I remember correctly, was around 90% or so. And so the risk of somebody being able to spread the virus after being fully vaccinated or having COVID-19 is definitely reduced. By how much is unclear. And it is absolutely possible for somebody to still spread COVID-19 after vaccination. And that's because we know the vaccines, none of them are 100% effective. There are people who still can get COVID-19 even after vaccination. And obviously those people then would be able to spread COVID-19. So it's not 100%. So, and that's part of why, even though somebody's been vaccinated, they're being advised to still wear masks and still stay socially distant, although those restrictions are easing somewhat. Let's see. Possible long-term effects of having this vaccine. Right, so we don't really know because it's also new. And for other vaccines that exist, long-term effects are almost nothing for vaccines that are just putting in a piece of a pathogen, something like putting in just DNA or putting in just the protein. There's pretty much no long-term effects that are known from that other than immunity to the infection. And one of the reasons that is that the vaccine doesn't actually stay in your body forever, right? For mRNA, it clears within a couple weeks, is only how long it actually stays in your body. The RNA, the vaccine, the DNA vaccines tend to stay in your body a little bit longer, maybe like a month or a little bit longer. And so the vaccine is definitely clears out of your body, eventually gets degraded. And then the changes that are left behind are changes to your immune system, which are just really normal parts of how your immune system functions anyway. Got exposed to something foreign, made some memory cells for later. That's happening in our bodies all the time. We get exposed to something foreign, we make some memory cells for later. So that part is really super normal. And the evidence for our long history of vaccination shows very little risk of long-term effects. The ones that have had some effects in the past, the vaccinations have pretty much exclusively involved attenuated viruses, where the actual virus that causes the disease was injected as the vaccine, and the virus was kind of killed before it was put in, but it didn't get killed all the way, and so it still had some way to divide in the body and cause some long-term effects. Like there are some really rare examples of people being paralyzed by the polio vaccine, for example. But it's thought from the science that we have and that we know that these vaccines are extremely unlikely to have any type of long-term health consequence. Okay. Why would an mRNA vaccine be used over a traditional vaccine? The major advantages of the mRNA vaccine are it's really easy to make it because you don't have to grow a virus in a cell. You can just kind of chemically synthesize the RNA, and then you package that in lipids, and bam, that's the vaccine. So it's really fast and easy to make it. And so I think that's probably the major advantage. One of the disadvantages is that RNA as a molecule is a little less stable. So that's why you might have heard about it having to be stored at these really cold temperatures, whereas the DNA vaccines or the protein vaccines can be stored at warmer temperatures. So that hasn't been as much of an issue. Um, does the coded DNA or mRNA change our own DNA? And if so, what are the ramifications of that? Yeah, so thanks for asking that. That's definitely been um, a worry that's been out on the internet by many people. There's absolutely no evidence of DNA or RNA vaccines being able to change our DNA. For the DNA vaccine, the DNA goes into the nucleus, but it doesn't actually interact with any of our own DNA. It's just hanging out there, and then it gets degraded after a while. It doesn't stay in our cells forever. For the RNA doesn't even make it into the same compartment that our DNA is in. So it has absolutely no way to interact with our DNA. And so, yeah, just the mechanism of how DNA could be changed doesn't exist for these vaccines. So the answer is no. The vaccines are not changing our DNA. Okay, why did they decide to make the vaccine out of mRNA or DNA? Yeah, so there are so many different ways that are used to make vaccines. The immune system typically responds the strongest to foreign proteins. And so some vaccines will just actually inject the protein. And actually I think it's the Novavax vaccine that is a protein vaccine. But if you're gonna just put the protein in itself as a vaccine, then you need a lot of protein in there to get a strong enough response. So it ends up being this really big shot, and you have to make a lot of this protein, so it's more expensive, et cetera. By just putting the instructions for how to make the protein, and those instructions come on the DNA or the RNA, that means that you can just put a little bit of this DNA or RNA in, and then your cells can just read that and pump out a ton of the protein for your immune system to react to. So it's kind of taking advantage of the way our cells work to turn them into little spike protein factories to turn on our immune systems. And then I know some vaccines are made of toxins or inactivated virus. Yeah, and so there are many different strategies that work. And basically biologists tend to just do whatever works. So they try a strategy and it works, and they're like, yes, let's use that. And that's why we have all these different vaccines coming out that are using different methods, and there are advantages and disadvantages to all the different methods. Okay, do you think there would be more mutations with mRNA vaccines compared to DNA vaccines, since most of the vaccines are made of DNA? So the vaccine itself is not going to mutate. The vaccine itself is made by the scientists in the lab and then injected in the body, and then it's used, and then it's degraded, and it never is seen again. And so those themselves will not actually mutate. The virus itself can mutate, and that's how we got the different variants, right? And I think what you're getting at is that RNA viruses are typically more prone to mutation than DNA viruses, and that is the case. And that has to do with how the DNA and the RNA are copied inside the virus when the virus is making more viruses inside of a cell. And so yes, we expect to see more mutations in RNA viruses than DNA viruses, but so far the data is showing that SARS-CoV-2 isn't, outrageously high in the number of mutations that it makes compared to other RNA viruses. There are definitely other, like influenza virus is way more likely to make mutations than SARS-CoV-2 is. So that's actually a really good thing, because we're hoping that these vaccines will be good for a little while and we won't have to make new ones all the time to try to keep up with variants the way we have to do with influenza. All right, the next question is, do you think COVID is here to stay? Can it ever be eradicated? Yeah, it's really hard to tell. some of these other related viruses, like some of the SARS viruses, did seem to sort of disappear. And so maybe there's some hope there, but those ones weren't quite as contagious and transmissible as SARS-CoV-2. So I think part of that is going to depend on how quickly we can mobilize vaccination and how long we can stick with our distancing and masks and other precautions. You know, if we can keep strong for another year or two and really make a concerted effort, then I think it could be possible to eradicate COVID, but it's really hard to tell if that's going to happen. And it's also hard to tell what might happen with additional mutations in terms of the virus variants. Do you know what else the vaccine is made up of? Yes. So I can actually, I'll copy and paste the ingredients of each vaccine into the chat so you can see. So no, it won't let me copy and paste that much. Maybe that's too long. Let's try it one at a time. Okay, so here's the Pfizer vaccine. And basically the Pfizer vaccine includes RNA. It includes a bunch of lipids, which are just a little package for that RNA that helps stabilize the RNA until it gets inside your cells. It includes some of these salts and some sucrose. And what the salts are doing is they're just making sure that the pH is right, the acidity is right. And what the sucrose is doing is it's just preventing the lipids from clumping and sticking together. The Moderna is a very similar formulation to the Pfizer. And the Johnson & Johnson has the adenovirus. It has the some of those same salts for making the pH correct, and that's about it. So they're actually pretty simple in terms, I know it doesn't look simple with all these crazy science words, but they're actually pretty simple compared to some vaccines that were made in the past that required preservatives and things like mercury containing compounds that people have been very concerned about. So none of that stuff is in any of these vaccines. Okay, so Paul, we have 2 minutes left. I don't know if I should stop.
Yeah, I was just going to comment on the time, and there's probably 5 or 7 more questions, which I think you've shared so much information for us. I just want a big picture. This is what happens when we begin to learn about something, right? We take that outside information and it begins to interact with the world that we know and the world that we've come to understand as we've been growing and learning. And that intersection right there is learning. And so I want to encourage everybody. I think what we'll do is give a way for folks to log off who do want to transition. And if Dr. Fox has a little time, she can continue to answer a few questions. But I just want to give you that big picture. Just because you don't know something now doesn't mean that one day you won't have the way to learn that. And as her experience, her trajectory showed, right? Going through the community college system gave her a way to be exposed to a field she had never been exposed to prior. And look at her today, right? She's leading us in this understanding of something that's really kind of affected our whole society. So I do want to kind of take this big picture moment to present this, to keep learning, keep pushing yourself. If you're given an opportunity, especially to go to a STEM lecture or to take a STEM class, maybe it's outside your comfort zone, but that's why we provide the network and the resources we do here at COM. We'd like to give you the comfort, the flexibility, the encouragement, even the resources to take that little step to explore something that you may not have considered. And who knows, kind of like Oda shared at the beginning, right? Who knows, you may find your passion. You may find a field of study that you never would have considered, changing the trajectory of your life. And I'll point you back to the other STEM speaker series speakers we've had this spring. Many of them have had these little moments they've shared, that there's just an influence from a mentor, influence in a class, influence from being together with others who had the same trajectory, that just helped to encourage them in that way. So maybe we'll go ahead and allow anybody who needs to log off at 1:30 to go ahead and do so. And we'll stay on for a few more minutes if Dr. Fox can answer questions. I think there's, thank yous coming in. Again, thank you so much, Dr. Emily, for the presentation. And look forward, we'll let you know when the next talk is. Drop by the link for our STEM learning community. If you didn't get it earlier, you can join right there and find out more. And otherwise, have a good afternoon. I think the question we were on was, do you have it in front of you?
Yeah. I do.
Okay, perfect.
Okay. All right. So thank you and goodbye to anybody who needs to leave. And I'm happy to keep answering questions. Even if everybody needs to log off, this is being recorded, so I can just sit here and answer questions for a while. Okay. So how will the pandemic impact the immune system of children that are social distancing and taking precautions to avoid the virus? I work at a preschool, and concern amongst my coworkers is that children are not able to build up their immune systems. Oh, so that's such a, I've had that same thought, because I have young children. I have a 3-year-old who is not getting very much immune exposure right now. We're stopping COVID-19 from spreading, but we're kind of stopping everything from spreading. And so I've had that same thought. I actually don't know if any studies are looking at that. I haven't looked into it myself, but I'm going to make a note to look and see if anybody's studying that. And I have honestly been making a bit of a concerted effort to roll my children in dirt a little more than usual to try to expose them to some microbes in another way. Okay. Why is the new variant affecting the younger population more than the original strain of COVID-19? So that's unclear, but one of the major thoughts is that a lot of the older population is vaccinated now, right? And so there's just kind of less spread in the older population in general now. And so younger population hasn't been vaccinated yet, and so they're just more susceptible. And as these new variants are more transmissible, they are kind of ripping through the available population that's left to them. Yeah, but that's really sort of a hypothesis at this point, isn't proven, but just one idea about why that might be. Are the symptoms the same on our pets that were exposed? Should I let people pet my dog? So pets don't have the same symptoms as us. My understanding is that some pets can get the virus. It's pretty rare. And when they do, their symptoms are different. I don't have a strong veterinary medicine background, so I don't understand that quite as well, but I don't think there are any cases of pets having severe disease or dying from COVID. If anybody knows differently, then please let us know. Should I let people pet my dog? I think that that's really sort of a personal risk decision, right? We're all making these personal risk decisions all the time in our lives right now. It's like, is this too much risk? Is this too little risk? So letting somebody pet your dog is probably pretty low risk, but it's not zero risk. There's some risk involved in there of, oh, maybe they have some COVID on their hand, some SARS-CoV-2 on their hands, and they rub it on their dog, and then you touch your dog, and then you touch your nose, or maybe just that interaction of them coming close enough to touch your dog puts you in the airspace with the person would probably be the more relevant risk. Both of those are probably really low risk, but not no risk. So it just kind of depends on where your personal risk tolerance is. I have not been having people pet my dog. so that's what, but my risk tolerance is pretty low. I've been very careful during the pandemic myself, and I know that not everybody is. And there's definitely a balance between the physical health protection and the mental emotional health of having interactions with human beings and being in the world. And that balance point is different for everybody. So I think it's kind of a personal decision. What is causing the blood clotting that we have heard about? Yeah, so the blood clotting can be due to a few different things. One idea is that if there's low oxygen, then that can cause the blood to clot. The other thing is that SARS-CoV-2 causes inflammation, and that inflammation can have a lot of effects on the body that can lead to blood clots. So narrowing the blood vessels or dilating the blood vessels, depending on which blood vessels you're looking at, and releasing immune factors called cytokines that can cause blood clotting. All of these different processes happening in the body when infection is occurring can lead to blood clotting. And whether it happens in one person or another is going to depend on your personal physiology and immune system. Are there any other ingredients in the vaccine that are likely to cause long-term negative effects? Nope. To the best of our scientific knowledge, the answer to that is no. Most of these ingredients have been used in other vaccines with no ill effect and have been used for vaccinations for many decades. But is it possible? It's always possible. Science isn't always right. And so there is some possible unknowns. For myself, it definitely is more of a risk to leave myself exposed to contracting COVID-19 than it is to potentially have a long-term health effect. And so that's the choice that I made. And probability-wise, it's very unlikely that there will be long-term negative effects from any of the vaccine ingredients. For those who get the second Moderna vaccination, there seems to be an increase in side effects. I've heard that because we don't have a good immune system. So others who don't have side effects, does that mean their immune systems are working correctly? Yeah, so this is a really hot topic that a lot of people have been discussing. Did you get side effects? I got side effects. I was out for days. Oh, you had nothing? Oh, is that because you're old and I'm young? Is that because I have this thing about me and you have that thing about you? And the side effects that we experience are actually separate from the immune response that we build. So the number of memory cells that we make and the strength of the immune response is not the same thing as the side effects that we experience. And the side effects we experience actually have a fancy science word called reactogenicity, which is kind of like how our bodies react initially to the vaccine. Whereas the immune response that we get to the vaccine of preparing that immune system is called immunogenicity. So they're actually kind of two separate things. And the reactogenicity can be altered by all different kinds of things. It definitely can be different in older or younger people, but there's also just an individual component of how strong is your innate immune response. So the innate immune response happens first. Things like fever and inflammation and kind of the very first line of defense, but it's not the one that's going to form your long-lasting immunity of antibodies and T cells that's going to be forming immunogenicity. And so those things have been shown scientifically to be separate. You know, just because you didn't have side effects, you still you still can be becoming immune and having a proper immune response. And you can kind of tell that by looking at the data, right? So for example, one of the mRNA vaccines, they're what, like 90-95% effective, but all those people didn't have symptoms, they didn't have side effects, and it still worked for them. So just by kind of loosely looking at the data, you can sort of tell that, okay, you don't have to have this strong side effects thing in order for the vaccine to work. Might there be a risk for mutations from the mRNA vaccine? There's thought to be no risk of mutations. I guess, do you mean in our own DNA, or do you mean for the virus? I think that, okay, I'll answer it both ways. So no risk of mutations into our own cells, not going to mutate us. In terms of, is it going to increase the mutations in the virus? No, it won't. So nothing can actually cause the vaccine won't cause more mutations to happen. But what can happen is that as the virus just mutates naturally, kind of randomly, if the vaccine is not effective against one of the new variants that mutates, then that variant can suddenly start spreading very quickly because the vaccine isn't working against it. And so that might seem like, oh, the vaccine created this new variant, but it's not really creating it. It's just that the variant isn't susceptible to the vaccine. Okay, I think that was the last question. If anybody wants clarification on any of my answers or anything, I'm
There's
happy.
one final question from Dr. Everett down at the bottom.
Oh, there is.
Is there anything to the notion of limiting anti-inflammatories in advance of getting vaccination?
So maybe. The answer is maybe. It is advised to not take anti-inflammatory drugs like aspirin, ibuprofen, Tylenol, all of that stuff before you get vaccinated. But if you have side effects, there should be no harm from taking some pain relief medication to help with those side effects. There's some limited data in some other vaccines, not for these vaccines, but some of the other vaccines that are given during childhood that show a less antibody production in children that take Tylenol or similar before getting the vaccine than children that don't. And so that's kind of what this is based on. But those children, even though they have a reduced antibody response, the vaccine is still effective in the long run. Maybe it's, so it does reduce the immune response, but not to nothing, and you still do have protection after that point. And so because we don't really know about the current vaccines, it's just kind of being advised to not take it if you don't need it, just in case. But the evidence is showing that probably, if you already got vaccinated and you took ibuprofen when you went, you're fine. You're probably fine. Maybe you made a few less antibodies, but you're going to be protected from the vaccine overall. You don't need to go get revaccinated or anything. Suggestions for keeping a strong immune system before and after getting the vaccine. Yeah, so that's, it's all the stuff that you've probably heard before, right? Is eating healthy foods and exercising and taking care of yourself. I know that there's been some talk about various supplements, things like vitamin D and vitamin C and zinc and those types of things. There's no hard evidence for that being protective to take supplements to protect you as for either COVID-19 infection or for vaccine side effects. There's some small studies starting to show that people, especially people of African descent or African American people, that if they have higher than normal vitamin D levels in their bodies already, that correlates with being less likely to have a positive COVID-19 test. So that's like brand new data that just came out in March, that's still being explored. And is that um, actually a causation or just a correlation? Is that because um, people who are more able to afford vitamin D supplements are also more likely to be educated in social distancing and mask wearing? Like it's hard to know if there are confounding variables or not, but there is some research kind of supporting the idea that higher vitamin D levels goes along with less COVID diagnosis, but nothing causative has been shown yet.
And Dr. Emily, I'm going to have to run. I know there's a
Okay,
couple more questions. Maybe I can transfer the hosting ability over to you
sure,
if
that's fine.
you can wrap up. Cool.
Okay,
Thank you again. Really
yeah,
appreciate your
yeah,
time.
thank you, Paul. All right, so the next question is, what about tetracumin SR as an anti-inflammatory? I have no idea what that is. So you know more than me, whoever asked that question. So I can write it down and take a look at it, but I don't know the answer to that, which I love. So one of the things I always emphasize as a scientist is when somebody asks you a question you don't know, that doesn't mean you're stupid. That means that you're a scientist and you're about to find something out. Um, and so that's something I always try to keep in mind is like, oh, I don't know something. Yes, I can be a scientist and try to learn the answer. So I can look into that. Tetracumin SR. Thank you. Why might children have been excluded from getting the vaccine is the last question I see here. So I think there were two reasons that they were initially excluded from studying the vaccine in children. The first is that the statistics show us that children are less likely to be severely ill from COVID-19, and so they weren't as much of a priority population. And then the second reason is that the immune systems of children are a little bit different than the immune systems of adults because they're still developing. They tend to have a much stronger innate immune response and not as much of an adaptive immune response. And that's still developing. And so they have to modify the vaccine formulation, usually decrease the dose and try to find a lower dose that's still protective. And so that makes for kind of just a different study. And then the other thing for children is just, we as a society tend to be protective of our children. Those are our babies. So when there's a new drug or treatment of any kind, we usually want to try it out on people who are consenting adults before we try it out in children, if possible, just to make sure that there's not some horrible effect that we didn't anticipate. So I think those are probably the main reasons. Somebody said, how can we get the recording? So the recording will be posted on the STEM Center page. If you go back up to the very top of the chat, I can copy and paste this in again at the bottom. So this is the STEM Center page on Canvas, and you can actually join this course. It's open to anybody, and then you'll have access to all of these talks, and all of the recorded talks can be found on that page. Okay. All right, I think we'll go ahead and wrap things up. Thank you all so much for coming and sticking around a little later, and have a wonderful rest of your week.